Do what on exam day?
Another extra video on exam day.
That's what Sherry would like us to do.
I think you should.
So we'll see. We'll see. All right. So we left off with talking about our sarcomere, and we're going to tie up the information. And I said that we would review and fill in the notes actually of the diagram of the sarcomere. So we said it with verbiage, but I didn't have the notes filled in for you guys of the actual what's actually happening. So when we talk about a cross-bridge attachment, so this is all talking about this diagram that we have up here with the sarcomere. And somebody had asked just a little bit ago about kind of how to segment the parts and how they all work together as far as the nerve has to stimulate the muscle cell first, and there were certain order of sequence of how that happens. And then also then the muscle cell actually getting the calcium out of the sarcoplasmic reticulum into the sarcoplasm, so inside of the cell, where the calcium then can do this sequence of events here with the cross-bridging with actin and myosin. So we'll go through these details. You will hear me repeat everything that I said again, and I highly, highly, highly recommend that you guys don't just look at your notes, but write like on a separate piece of paper all of the steps, the order that it happens, because remember, if something else happens or you skip a step, the other thing couldn't have happened. So it's very sequential, it goes in an order, and it can't happen any other way. So see if you can write down the steps, then go back and check your notes and see, did you get all of the steps? So that way you understand, and just recite it like a story over and over again. So let's see. All right. So cross-bridge attachment. This is where calcium becomes available. So it binds to troponin. So this is where we're in this upper picture right here that I will draw a circle around. This guy right here where I said calcium looked like the little snowballs. Troponin was the blue snowman looking guy. Calcium binds to troponin. Troponin then pulls tropomyosin out of the way. That's the green string. Okay, and then the little red balls are the actin, so the little cranberries if you want, because cranberries go with Christmas time, right? So when tropomyosin is pulled out of the way, it exposes the myosin binding site on the actin protein. Okay, so the string gets pulled out of the way, and as soon as that happens, myosin just clunks right into actin and they attach to each other. They bind to each other. If tropomyosin is covering the binding site, there's no interaction going on between actin and myosin. It's blocked. Okay, so now we've unblocked it. Myosin can touch it. So that's what the cross-bridging is. You're making a bridge between actin and myosin. Then we go down to what's called the power stroke, which was the second picture here. As soon as the binding of myosin head happens to the actin, it does like a flex or bend, or I like to think of it like a ratchet. It's like, right? So it flexes in. In which direction is myosin pulling actin? Towards the center of the sarcomere, towards the M line, right? Towards the center. And I'm going to redraw sarcomere for you guys as well in a second. So that happens, and as soon as it had bound, it also releases the ADP and phosphate, so adenosine diphosphate and phosphate from the previous contraction. So the previous contraction's all the way at the end of the last one is what they're saying, that ADP and a phosphate, that's what's now getting kicked off for the power stroke. So when that gets kicked off, that's what actually causes the ratchet. So myosin attached, ADP and phosphate gets popped off, and it ratchets the head in. So both of those things kind of happen really fast, and so that's why they say together that's the power stroke part. Okay. Then you'll have what's called the resetting of the myosin head. So this is where ATP comes in. So now we're at this part of the story. ATP comes in, because what do you think ADP and a phosphate, when it popped off, is going to end up doing? It's going to find somebody that'll put them together and make ATP, right? ADP plus phosphate gives you ATP. So now we have ATP coming in. As soon as he binds to myosin, that's what allows myosin to let go of actin. It kicks him off, and it resets the myosin head. So it ratcheted in on the power stroke, ATP attaches, and it makes it let go and resets the head back. Okay, so ATP splits into ADP and a phosphate, causing the myosin head to release and reset for the next power stroke. So ATP has to attach. When it does, ATP will break back into ADP and a phosphate, which is the one that gets kicked off later, because the cycle just keeps happening again and again until you run out of certain things. So this is where ADP and the phosphate, ATP becomes ADP and the phosphate, resets the myosin head, you've let go of actin. Now you're ready to start your cycle all over again. And if you want to think about it, think of it as kind of like myosin are like the people playing tug of war at two different ends of the rope. Okay? No shortening occurs of myosin. Myosin stays the same length. Actin stays the same length. But all you're doing is you're pulling the actin rope in towards the center, and when the contraction ends, you let go of the rope and it spreads back out. But it's not bunching up, it's just overlapping with the myosin. Okay? So same length on actin, same length on myosin. They're protein strands, they don't change length. They just slide over each other. And that's why they call this the sliding filament theory. So, all right. The other thing is they're showing you a picture of this one is a relaxed sarcomere. Okay? So you can tell the difference between this guy in the middle here is a picture of a contracted sarcomere. Do you guys want me to erase all my circles on there so they're not so confusing? Maybe? No? Okay. So contracted, so myosin, if you look at the length of one myosin protein, it's the same length in the contracted one. If you look at an actin protein, here's two red actin proteins from either side, they're just overlapped, but they're the same length. So the people playing tug of war, it's just like you're just pulling the rope in, but everybody, so the myosin actually has multiple myosin heads on one strand. So it's kind of like everybody's little hand or head is going like tug, 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 let go, grab another, let go, grab another binding site, let go, grab another binding site, over and over again until you've pulled it in and contracted. And then you'll stay that way until you decide that you're going to relax. And then all the little myosin heads go, okay, we'll totally let go now, and nobody's going to hang on. So it's happening at multiple places all at the same time. When we talk about this one cycle, we're saying like one myosin head attached, and this is what happened. But this is like 5 million little myosin heads attaching to all of the open actin binding sites for myosin that are tugging it in. Okay. So I was trying to show, did I draw this picture last time right here where I had the relaxed sarcomere, and then we have the A band is across here, yes? And then what was the, that's not all A band, I shouldn't have done that. I lied to you. You guys have to catch me on this, right? That was just saying one sarcomere. All right, let's draw what a sarcomere is doing. Let's see, do we have room? Oh, and then, sorry, at the end, removing calcium. So we have to get the calcium back into the sarcoplasmic reticulum, otherwise you won't be able to stop the contraction at some point, right? If calcium is there, the binding site is available. So we got to get the calcium out of there if we want to relax our muscle. So we have to pump it back in, and when we say pump, what does that imply to you?
Energy.
Takes energy, so we're going to need ATP to pump calcium because it's still in a higher concentration in the sarcoplasmic reticulum. So you're pumping it back in against its concentration gradients, shoving it all back into the sarcoplasmic reticulum. Once you get calcium out of the sarcoplasm back into the sarcoplasmic reticulum, which remember, it's like endoplasmic reticulum, it's inside your cell, then troponin doesn't have calcium attached to it anymore, so it releases tropomyosin, which then the string kind of covers back the binding site on actin. If the binding site's covered up, myosin heads can't touch actin anymore. So there's no interaction going on between them when that happens. So muscle relaxes because myosin can't bind to the actin anymore. Now, if we drew a, let me put a Z disc here and a Z disc here, and I'll just, you guys can use different colors or not. Here's an actin filament and an actin filament. I probably shouldn't have made it that long, right? And another actin filament and another actin filament. And I'm going to do red for a myosin filament. Okay. And so myosin has the little myosin head sticking up. And I'm just drawing a couple, but they're kind of all over this thing, right? but it's not touching actin because I'm going to draw something blocking it in just a second. So let's do neon teal. So this is pretending that you have tropomyosin covering up the binding sites, right? So if tropomyosin is covering the binding sites, the sarcomere is relaxed, okay? Now, if we wanted to have calcium released from the sarcoplasmic reticulum, and we are going to contract, so here's our Z disc again. Here is our myosin heads, or sorry, not heads, but the and actin is going to be blue. When we have calcium present, okay, then our myosin heads, tropomyosin got pulled out of the way, so I'm not going to put the teal stuff on there anymore. The myosin heads can now attach to the actin, right? And then what are they going to do when they contract? The power stroke, it's going to ratchet it in towards the center. So, come on. And let's see, we do our Z disc. So our Z disc will come closer together. And our actin is going to overlap because it got pulled in, but it's the same length as it was before. And our myosin, okay, so when it pulled it in, I'm kind of making my heads look like they've ratcheted in and they're still stuck in this position. So can you see like it went wunk into the center and it stuck there for a second? What does it need to do to let go of the actin? We need ATP, right? As soon as ATP attaches to the myosin head, ATP then splits back into ADP and a phosphate, and that causes actin or myosin to let go of the actin and to reset the head and relax back out. So then you end up looking back like the first picture again, right? And if it, keep in mind that this happened many, many times to tug it into the center, and it makes for a pretty smooth contraction because it's like all the little hands are like pulling in like this, and then they just let it go, and then they all pull it into the center, and then they let it go. So it's not like one guy goes because if different ones are grabbing and pulling it in, everybody's kind of working together, so it'll be a smooth pull in. All right, so you guys know that the A band is the dark band, the I band is the light band. Okay, so A band is where myosin and some actin, so it's the length of myosin. This is the A band, right? So same length on this one because we hadn't contracted yet. What about this one? Is the A band the same length or shorter? It's the same because it's the length of the myosin, but it's the dark band because it's a combo of both actin and myosin overlapping each other. So it's not going to be as dark in this area, but it'll be dark through almost the whole thing on that bottom one. But the length of the A band did not change because it's the same length as the myosin protein. And myosin and actin don't change length, so that's why that's the same. And then you guys know that the I band was the length of the just actin. So if, see how like along here, this is where the edge of the edge of myosin was right there. Let me see if I can erase that first set of dots I did. Okay, can you guys see that? So if this is this area would be the I band until like wherever the next myosin was. Okay, so the Z disc is in the middle of the I band. Okay, and so what happens in here is, will the I band change length when you contract? It does, because if we had myosin and actin overlapping in here, now your I band is only here. Because it's only actin for I band. Because if it's actin and myosin, it's darker because less light could pass through two things of protein, right? So keep that in mind. Don't just assume an answer, like redraw a sarcomere when you guys get your exam so that you don't get tripped up on whether an A band or I band changes length or anything. What about the H zone? Do you guys know where the H zone is? H zone is myosin only. So this part was the H zone. Does that change length?
Yes.
It does, because down here your H zone is only that long. It's where, that's an I, that should be an H. I can't draw today. My goodness, what's my problem? It could fully disappear if you contracted so hard that the actin filaments completely touch each other. It's possible.
Looking at the diagram we have with bones.
That, yeah, oh, yeah, so like, come on. No, oh my goodness. This one right here, they're showing no H zone because the actin is completely overlapping and covering the myosin all the way through. So you would not see an H zone on the fully contracted one. But we don't always contract that hard. But you guys have picked something up before where you had to really contract your muscle hard versus, you know, knowing like I just have to pick up a pencil, I'm not going to use full max strength, right? Because I'm anticipating my muscle memory and what my brain knows how much a pencil should weigh. But if this was like some weird planet and that pencil weighed like 500 pounds, that would be really bad. I'd be like, oh my God. And then I'd like contract harder to try to lift it. So we don't always max contract, but you can. And if you did, you would lose the H zone, yes. And the M line would still be there, but you don't really visualize the M line so much. It's more of a, that's what holds myosin kind of centrally, okay? So that's why I wanted to talk about the I and the A band after we had talked about what's happening with the sarcomere, so you guys could kind of visualize what that contraction actually looks like. And then it would spread back out. Now it's possible to overstretch something, okay? So what if, on this picture here, what if we overstretch our muscle? So this would be like somebody taking my arm and stretching it even more and then telling me to try to contract against what they're doing. Do you think I would be able to contract? Well, let's draw a sarcomere and see what happens. So this would be overstretched. So let's say here is my Z disc. We still have actin coming across here. We still have actin there, same length on all those guys. And then we have myosin here still in the middle, but if I'm overstretched, can my myosin heads even reach my actin? That would be the problem. Even if I get calcium in there from the sarcoplasmic reticulum, if my myosin heads can't reach their binding site, no contraction will be able to occur. Or if you barely get one guy that goes, wait, I can almost reach you. Hold on, I got you. You know, you would have a much harder time trying to contract that muscle than one that's in a normal average position, which would allow you to have the best possibility of contracting versus what about a muscle that's already contracted? How much more force would you be able to generate if it's already contracted and now you want to start from that point? Not much more, right? It's already shortened and you don't have much more that you can overlap after that point. So this guy in the center would be your best bet to have the best amount of force to overlap and cause the best contraction. It doesn't mean you can't contract on the other two. It's just a lot more difficult and it's not as efficient. Okay, so we'll talk about that again in a second, but I wanted to point that out. Good question about the H zone. And now let's talk about rigor mortis. So, do you guys know what rigor mortis is? You have died, but your body doesn't know it yet. So, things are still occurring inside of your body. So, let's say you expire, you pass away, but other metabolic processes on the cellular level are still occurring. Okay. And what happens is it takes about, so if you had contracted, let's say, right, calcium is out of the sarcoplasmic reticulum. If calcium is there, you have a myosin head that would be attached to the actin, correct? Okay. And then you would hang on until ATP comes around to make it let go. Yes? So if, let's say, though, after you have expired, it says cross-bridge detachment is ATP driven. So if after a time you run out of ATP, you can't do what? If you don't have ATP, what happens to myosin and actin? Myosin is stuck to actin then, okay, until ATP could be broken down. So when you first die, you are no longer breathing and your blood is no longer pumping. So if you're not breathing and blood can't transport oxygen to your various tissues, you run out of oxygen. You run out of oxygen, you won't be able to do later what you guys will learn is cellular respiration, which is the ability to break down your glucose and make the most amount of ATP. So if you stop making ATP because oxygen's no longer available, now your mitochondria is not making ATP. Now you have no ATP to make your myosin head let go. So it takes a few hours for that to happen, and that's why you'll start to stiffen in about 3 to 4 hours. Okay, that's when the start of the stiffening happens. So no ATP to power the calcium to put that back in the sarcoplasmic reticulum either. So you've got calcium sitting out there where you can't get it back in, and you have no more ATP either to make your myosin head let go. So those are both the things that end up stiffening you up. This peaks after about 12 to 15 hours, give or take, and then it starts to dissipate in the next 24 to 48 hours. So then you start to soften up again. Okay, so rigor mortis is the term that they say, you know, like when you stiffen up and you're really firm and stiff, and then you start to soften again, and then you'd be more pliant and stuff. So the reason you start to soften up is after a period of time, you still have enzymes, proteins that are still doing their job. They don't know you've died. They don't care. They're there to do a job. They find the thing that they bind to and they break things apart. So after a while, the ATP might get broken down, so the myosin head would then let go, right? But you still can't get calcium back in, so that's just floating around. Other things aren't functioning well now, so now your lysosomes start doing autolysis, self-chewing up, okay, self-breakdown. So your lysosomes start breaking things down, and now you don't even have whole cell parts anymore, and other functions aren't happening. And so that's the other part of softening up. You're making yourself like a jelly mess inside, right? Yeah. So autolysis by use of, because what's inside of lysosomes? Tons of breakdown enzymes, right? This is inside the cell. When you had like a mitochondria that was starting to die and fall apart, your lysosome could attach to that other vesicle of bad cell parts, and then it would dump its enzymes in there and start breaking it down, and then you can recycle the parts. The problem is you're not recycling anything at that point. Lysosomes don't know that, but you can't recycle anything because you're not alive to utilize recycled parts anymore. Okay, so does that make sense what happens on why you would stiffen up and then why you would end up softening again with rigor mortis? Yes?
When it dissipates, is
Uh-huh.
that from the very, very beginning when death occurs, or is that from after the peak?
I think that's after the peak, because in previous notes it was like 48 to 60 hours. So I think the whole process was give or take 48 to 60 hours. The other thing to keep in mind with rigor mortis is your environment would make a difference on how fast or slow this would occur. So if you, say, got frozen outside when you died, like an avalanche, like those people that died recently in the avalanches in Washington. So if you were cold, cold slows down all the processes, right? So this would take longer than 3 hours for the stiffening to occur. It would take longer than the peak 12 to 15 hours. It might spread it out to 20 to 30 hours or something. And then finally, when you start dissipating, if it warms up enough, then you might start melting away inside, not outside. If, let's say, you were down in Arizona out in the desert in the middle of summer and this happened, the 3 hour thing, what do you guys know about enzymes and temperature? Temperature, if you have more heat, speeds things up. Things are more likely to interact with things. So you would probably stiffen up faster than the 3 hours potentially. You would have your peak maybe at 6 to 10 hours maybe, and then dissipate maybe closer to 24 or less hours potentially. So depending on the environment, and they use this in forensic science, they take the temperature of what was going on when the person possibly perished, and they can pinpoint how long they think the person may have been there before they were discovered, and because they can use all of this information and glean that. So yeah, so roundabout numbers on there because again, it depends on the environment and what's going on. All right, so what else do I want to tell you? I talked about the sarcomere. So if we review, let's see, regulation of a contraction, nerve stimulus, and the neuromuscular junction. So what I'm saying here, you guys have already heard. This is like reviewing that, exactly. So motor neuron is an autonomic, somatic motor neuron. Somatic is because you are telling it what to do, because skeletal muscle, you control when you want to move it, right, for the most part, okay? So this stimulates a muscle cell. It does this by the neuromuscular junction, neuro for nerve, muscle for muscular junction. So where the muscle cell and neuron join, and this is one per muscle cell, and the events of the action potential propagation. Propagation just means like if it touched the cell here, it propagates or passes it along the whole cell membrane really fast, okay? So voluntary impulse from the brain. You say, I want to pick up my coffee mug. Voluntary, I want to pick it up, right? And electrical impulse from the brain, the nerve zaps that muscle saying, do this. If I contract you, you'll be able to lift this, right? So this causes you to exocytose acetylcholine. That is your neurotransmitter. The receptors on the motor end plate, where are those located? Motor end plates are on the muscle cell membrane, okay? So that has a binding site for acetylcholine to attach to it. This, when acetylcholine attaches to the motor end plate of the muscle cell, it opens the sodium gate, and so sodium floods into the cell. And did we talk about last time how that will change the voltage on the cell? Okay, so you get that voltage change. That's what tells the sarcoplasmic reticulum to open the calcium gates. So if you don't open sodium gates to flood the whole muscle cell, you can't open the calcium gates from the sarcoplasmic reticulum. Okay? Calcium then floods into the sarcoplasm. So what happens is all of this is high to low concentration. Sodium's higher outside the cell, so that's why it floods into the cell when the gate is open. Calcium is higher inside the sarcoplasmic reticulum, less in the sarcoplasm. So you open that gate, calcium wants to flow out to where there's less of it. And this is all from, earlier chapters that you guys learned that part. The electrical impulse propagated along the sarcolemma. So the voltage change on the cell, what it's saying is went along the whole sarcolemma, the whole muscle organ felt it. Okay, or that cell of the muscle organ felt it, I should say. That's what tells you to release the calcium. In the meantime, remember in that synaptic cleft between the synaptic knob of the neuron and the sarcolemma of your muscle cell, you have acetylcholinesterase, ACH, ACE, right, or E is what they put there, is rapidly chewing up acetylcholine into acetyl and choline. It's breaking it into two pieces. So acetyl and choline separately don't stimulate anything for you on that muscle cell. Because remember we said anytime we have receptors, they're very specific where a protein can attach or not. Something that looks slightly different won't attach, it won't cause anything to happen, right? So acetylcholinesterase is chewing up acetylcholine. If you take away the acetylcholine, then we are stopping that voltage change on your muscle cell. But while you want to sustain this contraction and hold your muscle, your mug up of coffee, right? If you want to keep contracting, you keep telling the nerve, keep sending acetylcholine there. And you have more acetylcholine flooding that synaptic cleft than acetylcholinesterase breaking it down. So it's there too, but it's not breaking it down as fast as you're flooding acetylcholine. But at some point you go, you know what? I'm getting fatigued and tired. I don't want to hold my mug up anymore. I'm going to put it down, stop the contraction. So now I'm no longer, my nerve said, okay, stop the acetylcholine. No more acetylcholine's being put in the synaptic cleft. Acetylcholinesterase now has time to finish chewing up what was left in there. And as soon as there's no more acetylcholine, no more contraction, because now you have no more what? No more voltage change, thus no sodium going in. No sodium going in, now calcium is not going to be released from the sarcoplasmic reticulum. You guys, everything that I keep saying to you and repeat myself and I go forward and backward in the story, that's what you guys need to do to get this under your belt. Keep repeating the whole story, even though it seems really silly and ridiculous. And you're like, I don't want to say it again. Say it again and again and again. Write it down again and again and again. Okay? And then so when the action potential stops, acetylcholine rapidly is destroyed by acetylcholinesterase. Sodium gates close, causing the calcium gate closure. Cool. Does that make all sense, right? So now we have another lovely picture. And this is your somatic, I should go back to black probably. All right, so here is your axon. You have your synaptic knob here, all these little vesicles there. Those are your acetylcholine, your neurotransmitter that's being formed there. And then that's going to get released into the synaptic cleft. You're going to have your motor end plates that will recognize acetylcholine that are on the sarcolemma of the muscle. That will open up then the what? Sodium gate. Sodium then floods into your cell. So let's write sodium here. He then can flood into your cell. He causes then a voltage change. That then tells your sarcoplasmic reticulum, which is the blue network stuff here, to release calcium. So that's showing you all the little calcium balls here. And then what's calcium going to do? Attach to troponin. Troponin then moves tropomyosin out of the way. This allows myosin to now bind to actin. Okay, now actin will ratchet it in. ADP and a phosphate get kicked off. That's what allows the power stroke. Then ATP has to come in, attach to the myosin head to make it let go of the actin and recock the head so it can do it all over again. And as long as you have ATP and calcium, you can do a contraction. You run out of calcium or you run out of ATP, you get too fatigued, you can't do anything, right? You become weaker and weaker, you can't do the contraction. So you either have to rest or you've died and then you'll stiffen and then you'll loosen up again, right? Yes, okay, awesome. Guess what? This latent period is the period of time between the action potential, meaning when sodium came in and said, okay, I'm changing the voltage on the cell. It sends an action potential across the whole sarcolemma of that one muscle cell. The action potential, there's another part of the story also. So when you want to stimulate the nerve, it actually will have calcium, not from, I mentioned this briefly last time, calcium not from the sarcoplasmic reticulum, because that was inside the muscle cell. This is outside of both cells. Calcium is what actually tells you to release the acetylcholine, what am I trying to say? No, not the esterase. Acetylcholine. Oh my gosh, I can't say it now. See what happens when you say it 5 million times fast? Acetylcholine. I'm trying to say acetyl-CoA, which is something else. Acetylcholine, ACH. All right, so calcium actually comes in and stimulates that nerve cell. It causes an action potential here that causes you to make the acetylcholine, and that will then get exocytosed to the muscle cell. All of that up until the point where you actually shorten the actin and myosin fibers is what's called the latent period. So from the time you're telling your nerve cell, I want to move something, to just before you actually contract and start pulling in the sarcomere is the latent period. That is only several milliseconds. Okay? So do you guys, if you want to put milliseconds into perspective, okay? So 1,000 milliseconds is 1 second. So this is just several milliseconds. So if it took you 100 milliseconds to do something, that's only 1/10 of a second. So this is less than that. So when we say this takes a little while to do this, it's really, really fast. Okay? So we'll talk about that again in a second, but that's the latent period. It's just several milliseconds, which is not very long. Talking about contraction of a whole muscle. So talking about the motor unit. So motor neuron plus all the muscle cells it innervates. So let me go back up to that picture that we talked about in the last lecture. Do you guys remember this picture right here? So we have our nerve, and some of the neuromuscular junctions, here's one, and it has like a little head here, a little synaptic knob. Here's another one. Notice how these are on different muscle cells. And then I could have another axon nerve fiber that can do the blue one. So it's innervating this muscle cell and this muscle cell over here. So the blue one only has, just for this picture and this purpose, it only has 2 neuromuscular junctions. 2 different muscle cells being innervated by one nerve is what we're saying. And the red one that we can see, we'll just count the 3 that are obvious. So the red nerve has 3 neuromuscular junctions, NMJs, okay? So, and they're each also on different muscle cells. So the red one is stimulating one more muscle cell than the blue nerve was, right? Okay, so that's what I wanted to point out on here before we go back down to this picture, because now we're talking about that. Okay, so all or none. When you activate a motor unit, okay, all of the muscle cells it controls will contract. So that red one that was touching 3 different muscle cells, if that nerve is stimulated, all 3 of those muscle cells will contract. The blue one only was touching 2 muscle cells. So if that blue nerve is stimulated, then 2 different muscle cells are the ones that are going to contract. Okay, does that make sense? And if you reach something that's called threshold that you're going to see in a second, if you reach that point, it means it's going to happen. You're going to get the contraction. If you don't have enough voltage change to reach that threshold, you won't get a contraction. Okay, bless you. So that's why we say it's all or none. If you reach threshold, the one that nerve is activating, anything else, any other neuromuscular junctions it had, will all contract with it. Okay, so that's the motor unit, one nerve innervating multiple muscle cells, depending on how many junctions it had. Okay, so each muscle possesses at least one motor nerve, otherwise it wouldn't have something to tell it what to do, but it may contain hundreds to thousands of neuromuscular junctions for one nerve. So that nerve can contract and talk to a whole bunch of other muscle cells. And each cell has to have at least one neuromuscular junction. Otherwise, it wouldn't have something telling it what to do on skeletal muscle. So one motor neuron may have 4 to 150 muscle fibers that it controls. And then it wants you to know which one has the best control for force generation. Okay, so for example, if you go back to that first picture before you read muscle A and B. So let's go back to our red and blue fiber here. So each knob is a neuromuscular junction. The knob is on the neuron side. The motor end plate was on the muscle side, right? Synaptic cleft was between them. That's a neuromuscular junction. So from this picture, motor unit 1, which we'll call the red guy, and we'll say motor unit 2 is the blue guy. So motor unit number 1, the red one, okay? So from this picture, motor unit 1 is showing more neuromuscular junctions than the motor unit number 2 of the blue guy, right? Okay, so the force contraction for this muscle organ, do you think it would be better to have the red neuron with 3 neuromuscular junctions, or would you get a better contraction with the blue one with 2 neuromuscular junctions? The red one. Okay, so keeping that in mind, coming back down again, have I made you guys dizzy with the notes yet? Probably. Okay. Please don't have a seizure. Okay. So, example for part C. Which has the best control over force? So you have muscle A has 1,000 motor neurons, each attached to a single muscle cell. And you have muscle B, which has 1 motor neuron attached to all 1,000 muscle cells. So what it's saying is muscle A is a 1-to-1 connection, right? It's saying muscle B is a 1 to 1,000 connection. Which one has better control? Which one has better control? A would, because you have one nerve telling one muscle cell what to do. Okay? So it would be a little more finesse. You don't always want to activate all of your muscle cells all at once. Otherwise, you might get that max contraction and get more than you bargained for, like smack yourself upside the head with your coffee mug, right? Because everything contracted all at once instead of, I will just recruit a few of my muscles and I can decide to delicately pick up a pencil and delicately put it down instead of whonk, whonk. Kind of like Men in Black. Do you guys remember Edgar in the Edgar suit and how he moved around, right? So I kind of think of it like that. So A has the best control or finesse or force generated because it's a one-to-one connection. B will contract maximally though because it would be a lot less control. You can't just activate just a few muscle fibers and do a graduated contraction. It just like all or nothing, bang, bang, you know, and you smack yourself in the head. So let's see. On this muscle twitch, this is a diagram. So the muscle response, so a myogram, myo for muscle, it's just saying, how do we record this on a piece of paper and show you what's going on, right? So you guys probably had one of these on your fills that's due this week? This week? Yes? Okay. So the response of a whole muscle. So what they did was the whole muscle, so one muscle organ, so this might be your bicep muscle, or you could say your rectus femoris, because you guys are doing lower extremity this week, right? So this is one muscle belly, one muscle organ, the whole thing, not a cell, multiple cells are in here, okay? And then they suspended it between like the bar here, and then they put a little weight on it so that it kind of gives it a little tension, okay? So that you could also measure if the muscle contracts when you zap it with an electrode. So this is like your nerve telling you you want to move your muscle. Then you can measure the length of the muscle. Did it shorten? And it's that perceptible shortening is what we call the twitch. Okay, so let's see. So we have a skeletal muscle. I talked about it being attached. If the zap is strong enough for us to reach threshold, it will cause a contraction or muscle shortening or a muscle twitch. And then you could record it on this myogram, okay, this guy right here. So this is amount of time that had to pass and how much muscle tension or how much it's contracting or shortening, right? So threshold is the minimum stimulus necessary to cause a visible motion of the muscle, the shortening. Some coffee without pouring it all over myself. So the myogram is recording, and we have 3 periods that we need to look at. So here's myogram as the recording. It has several components. You have a latent period, a period of contraction, and a period of relaxation. Remember earlier I said the latent period is just a few milliseconds, okay? This is, again, everything that had to happen between the action potential reaching the synapse on the nerve side, telling it to send the signal and zap the muscle with acetylcholine, Thank you, acetylcholine. Oh, you guys will laugh because in 228 you're going to say acetyl-CoA tons of times, and then you're going to be like, that's where she got it from. Because I'm thinking about ATP and metabolic regulation right now. I apologize. ACH. I'm just going to say ACH from here on out so I don't screw it up for you guys. Okay, so the latent period was everything that had to happen between the action potential reaching the synapse and the initiation of the muscle shortening. So you didn't shorten yet, but it's the just before you're about to be able to shorten, okay? And how long was that latent period? A few milliseconds. The period of actual contraction, this is talking about the power stroke when we looked at the sarcomere picture. So this is where myosin head ratchets in, okay? That lasts about 30 milliseconds, okay? Not seconds, milliseconds. And then the period of relaxation is 100 milliseconds. And it says calcium sequestering. Sequestering just means this is when the calcium needs to get pumped back into the sarcoplasmic reticulum. This is where your muscle is relaxing in between the next contraction, okay? So it takes a lot longer to put calcium back into the SR, sarcoplasmic reticulum, than it did to actually do the contraction, right? Because it takes a while, you need energy, and you have to pump that calcium back into the SR. So latent period, just a few milliseconds, that's from here to here. The actual contraction of the muscle was give or take about 30 milliseconds. The relaxation period was a lot longer. That took you about 1/10 of a second to relax. That's why I was saying 100 milliseconds is 1/10 of a second. I was just trying to put it in perspective for you guys. Because sometimes people throw numbers out and you're like, I don't know what you're talking about. Now, hopefully that makes a little more sense for you guys. So, let's see, what else do we want to say? Now, knowing that information, we're going to look at what's called a graded muscle response. So, a smooth, gradual increase in muscle force production, okay? So, let's see. This is about control. So, how much of a contraction we get and how fast or slow do you want to do that contraction? That's what a graded muscle response is. And we do this by 2 different ways, either by doing stimulus frequency, which is a wave summation, or we can do it by more of a motor recruitment. You guys understand what I mean by motor recruitment? more of those neuromuscular junctions saying activate, stimulate it, cause another muscle cell to contract. Okay, recruiting more muscle cells. That's what that means. So let's look at stimulus frequency or wave summation. Let's see. Yeah, so this is 2 or more successive stimuli rapidly delivered. And a stimuli is that electrical zap. So if you want to talk about the picture up above where they artificially are zapping the muscle, or whether you're telling your brain says, contract, I want you to move now, okay? So the wave summation means how often do you stimulate it. So in this first picture for Twitch, they are stimulating it about every 10 stimuli per second, okay? So notice that all the contractions, the muscle tension are all at the same height. So it's like saying contract, relax, contract, relax. So the dip down is the relax, the contract is where it hits the peak. Okay, so that first diagram, we hit the muscle with 10 stimuli per second. It's good contractions. It has a full amount of time to have its full relaxation between the next stimuli or zap. That's a twitch. So you're seeing a little twitch going on with it. Doesn't mean you fully contracting your muscle every single time, but it's enough to get it to twitch. Have you ever seen, like, you hold your arm out and you see something like flutter and you know it's your muscle and you're like, this is really weird? That's a twitch, okay? It's like an alien underneath you. So the second picture, this guy here, this one is we increase the frequency, so we're hitting it about twice as often, so 10 to 20 stimuli per second. And notice that the contraction is getting stronger, okay? It still relaxes in between each one, but it's getting a stronger peak, so a stronger contraction, or almost contraction, if you will. And this is what's called Treppe, T-R-E-P-P-E, okay? The Treppe phenomenon, Treppe is German for staircase, so can you see it's a staircase effect? That's what Treppe means. Pretend like I can draw stairs, okay? That's Treppe. So a staircase effect, meaning also like a warming up effect. So you guys know if you exercise, you go to the gym or you run outside or something, or you're playing with kiddos and you start to get warm, your body warms up. The more you warm up, the more flexible your other tissues get, like the CT and the other stuff. The muscle warms up. What happens when you warm up things enzyme-wise? You can have faster reactions occurring. So that's why it's part of a warming up effect also. So you're making it easier and easier to get more of a contraction at that point. All right, so let's see, what else do I want to tell you? Told you all that. In the third diagram, this guy right here. So if you reach this plateau, so plateau is like when it levels off at the top there, and it's got a smooth top. This is called tetany. And if they had a really delicate instrument, it wouldn't be completely smooth. You would see still like little wiggles at the very tip of it. But, you know, when you step back and you look at it, it'll basically seem like a flat plateau or straight line there at that point. So that's what's called tetany. So the frequency of stimulation is so great. You're stimulating it so fast, right, that the muscle doesn't even have a chance to relax. See how it doesn't dip down to the baseline like all the other two pictures did? Okay, so you don't even get a chance to relax in between. You're stimulating, zap, zap, zap. And so then you end up getting like the tetany, like the full contraction. Have you ever had like a muscle cramp where it tightened up on you and didn't let go? You could think of it like that. Okay, it doesn't relax. It's not having a chance in between. So this also means if it's not relaxing, what is calcium not getting to do? Because we said during that relaxation period, what was happening to calcium? It was going back into the sarcoplasmic reticulum. If you don't have that relaxation occurring, calcium's not getting put back into the sarcoplasmic reticulum. It's staying in the muscle sarcoplasm. That's why you have that contraction still going. Calcium's still available. Sites are uncovered. Myosin still attaches. Okay? And let's see. So with tetanus, that's where we're saying the myogram, these pictures, the drawing of it, look smooth or plateau. Okay. Let's see. Motor unit. Where am I at here? Motor unit recruitment and summation, increasing the number of motor units activated or recruited. I like the term recruitment, but activated or recruited. And that will increase the force produced. So this is talking about stimulus strength, right? So if you held something out for a while, you will have some of those muscle fibers will fatigue after a while, right? If you didn't hold anything out, you could hold your arms out longer. But even just holding it out against gravity and the weight of your own arm, you start to get tired after a little bit. You'll just speed it up if you actually put weights on your hands, right? So while you're doing this, you have stimulated some fibers to be contracted and firing. And then after a little while, they're going to get fatigued. And so then you're going to recruit other muscle fibers in that muscle belly to then wake up and be zapped, and they're going to take over so the other ones can rest a little bit. And if you still have to hold it, then other ones are going to get recruited. Okay? Otherwise, it would be like this constantly. You can't even hold it up anymore. So you are able to sustain a contraction because you can do summation. Okay? Motor unit recruitment is what that's about. So the more motor units we turn on those neuromuscular junctions, if we turn more neuromuscular junctions on, the greater the force produced. Okay? So we turned on some motor units, and after a bit of time, they fatigue, so we can recruit and activate and turn on more neuromuscular junctions to let the fatigued ones recuperate or rest. Okay? Looking at the term isometric and isotonic, iso means the same. Okay? So it helps when you know what something means. Isometric, meter is like length. Okay? Tonic, you're talking about tone. So isometric means the same length, but it means the force changes. So this would be like if I stood in a doorway and I push against the doorway. I can't move it because I'm not strong enough to push wood, right? So I'm pushing against the doorway. That would be isometric. I could push, and increase the tone, but I have not changed the length of my muscle. Okay? The muscle length didn't change. It didn't shorten so that I could lift something up. That would be isometric. Isotonic would mean that you are holding the same force. So I've picked up my mug. I'm contracting a muscle in order to do it. I have the same amount of force just to hold it, but the length of the muscle had to change in order for me to pick it up, right? So there can be a sequence of events where these two terms can be related. So, for example, if I see this mug, or maybe this is a really heavy kettlebell at the gym, or maybe it's a heavy box because you just moved or something. And you go over, and when you first go to pick it up, you don't realize how heavy it is. So when you first pick it up, you are contracting your muscle to try to lift it, but if it hasn't moved, it is still a what contraction? Isometric, okay? As soon as I overcome the force of how heavy it is and I recruit more muscle fibers and go, okay, and I actually lift it up, now I've contracted and changed the length of the muscle to lift it up. Now it's become a what? Isotonic contraction. Now, if I want to put it down, I'm changing the length of my muscle. I'm relaxing the muscle to let it down. So now I'm doing what? I'm still changing the length of my muscle. So it's an isotonic relaxation. By the time I set it down but haven't let go, I still have some contraction there. That would be an isometric relaxation until I let go finally, correct? Can you see how they can be related? Depending on, so the actual movement, once you're moving something, now you're changing the length of the muscle to shift it and do something, right? All right, cool. And talking about muscle fatigue, let's see, what do we want to talk about on muscle fatigue? Oh, another thing that I was going to say. So if isometric is the initiation of the movement, but you haven't actually moved it yet, isotonic is where you actually start moving it, right? And you have to recruit even more muscle fibers if you want to move faster or higher or something like that, right? So isotonic might be things like jumping or throwing, and that's when the filaments are sliding if it's isotonic, right? Because you have motion going on. You're contracting, changing the length of the muscle because you're shortening the sarcomeres. Isometric would be the attempt to move something. This is where a cross bridge has occurred, but no sliding has happened yet. Does that make sense? You didn't shorten the sarcomere, so you've made a cross bridge. You have tension, but you're not moving. You didn't lift it yet. So no change in the length of the sarcomere. So you've got your cross bridge, but no sliding filament yet. I was just going to tell you guys that and point it out. So muscle fatigue, if we look at reduced ability or inability of the muscle to generate tension, okay? So, and this means even though you're trying to stimulate it and make it do it anyway, right? You're fatigued and tired, but it doesn't want to listen to you anymore. It says, screw you, right? All right, so let's see. I have cool things to tell you guys. There's so much to say. So little time to do it in, right? Okay, so everyone has probably felt this at one point or another, right? You chased your kiddo around or you ran up and down the stairs so much and you just can't quite do one more step up the stairs. It's too fatigued. Your legs feel like jelly or your arms are jelly from holding something for so long. That's the sensation. You're telling it, come on, just one more. I need to just get up one more step and I made it to the doorway, and your body says no. Okay, that would be muscle fatigue. Different reasons why this can happen. It could be due to glycogen. So your stores of glycogen could be deficient. So you guys know glycogen is your animal storage of glucose. We need glucose for energy. So if you have depleted that because you used it all up in the exercise or whatever activity you just did, and now you have no more of it, now you don't have anything to break down for more energy, then that could cause the fatigue. If, for example, so like marathoners, okay, or people going to do like triathlons or any of those kind of things, what do they do the night before? They carbo load, so they eat a bunch of pasta or potatoes or whatever it is that they want to eat. They load up on carbs, which is glucose. They're trying to preload their body full of glucose so that when they run out of their own glycogen stores, they might still have some there that they could utilize and might be able to go a little bit farther or longer than they would have without having done that. Electrolytes. So you guys realize electrolytes are salts. We kind of talked about it earlier in the chapters. So you could have an imbalance of calcium, sodium, or potassium. And if you don't have the right balance, you don't have the right amount of sodium outside of the cell and potassium inside of the cell, then you won't be able to do like your sodium potassium pumps, and the leak channels get all screwed up. And that could get so bad it's detrimental to your life, right? If people drink way too much water or get too dehydrated. So people doing these sports where they do like Ironman and stuff, and they don't drink enough fluids, and maybe they're drinking enough water, but they don't have enough electrolytes putting back in. That's why they'll drink like, not necessarily Gatorade, but some of those kinds of drinks to get those salts back into their body. Because if you get out of balance, you won't have things to properly function on the cell. O2 debt, so we need oxygen. If we run out of oxygen, so let's say if you were running so fast that you can't really breathe, you can't talk while you're running, you are out of oxygen, okay? And it's not getting, you're alive, you're getting enough oxygen in to be alive, but you're not getting it to the muscle that is actively needing tons of oxygen at that moment. So like your thigh muscles need tons of oxygen as you're running down the road, right? If you don't get oxygen to that area very well, then it's going to run out, and then you end up doing like lactic acid and things like that, and you start to get sore maybe. And so with oxygen, we do have in our skeletal muscles myoglobin. So hemoglobin is in your regular bloodstream bringing oxygen, but you also have myoglobin that's in the muscles. So once you run out of your systemic circulation where you're not getting it to that muscle, you'll start to use it from your myoglobin locally in that muscle. And then when you run out of that, then you might be so fatigued you can't continue until you take a break and replenish your stores of whatever it was, or all three that you might have been missing, okay? And let's see, what else do we need to know? That was cool. Force, velocity, and duration of the muscle contraction. So let's see, force is affected by various things like the number of fibers contracting. So the more fibers you contract, the more force and strength you would have, yes? If the relative size of the muscle fiber, so we're talking about the diameter of the muscle cell itself, okay? So if it's a larger diameter muscle fiber versus a skinny little muscle fiber, this guy's gonna have more force, okay? If the stimulus frequency, the more you're zapping it, right? So increased stimulus frequency would increase the force. You guys saw that on that diagram, right? The more we zapped it faster and faster and faster, pretty soon it couldn't even relax in between, and it just was totally contracted. And then we also have the length tension relationship. So this is how much the thick and thin filaments overlap. So this is where I was talking about earlier about a regular resting length sarcomere was the best one, right? And we said a contracted one really couldn't get very far because there wasn't as much room to finish overlapping. You had very little overlap, or that one's showing a totally contracted one, so it can't overlap anymore at all. And then the one that's overstretched where the myosin heads can't even reach the actin sites, that guy's not going to contract very well. So they just did like the length of the sarcomere itself, and they're saying that this and this area between here and here is your optimum length of a resting sarcomere that would give you the best forced contraction. If you're already contracted, so this would be at 100% of the contraction if you reach this guy right here. If you're already contracted, you're not going to have much further that you can go. So it's not going to change the length of the sarcomere much because it was already shortened, and you won't get 100% contraction. Maybe you get 75% of the contraction because you didn't have as much that you could contract anymore. Or you only get 25% more maybe or something. And if you're overstretched, I mean, look at this poor guy. If he was landing here, he's also getting not as much if it can grab on, right? If it can't even grab on, you're not even going to get a contraction. But so I wanted to point that out. And when we're talking about the muscle diameter, for those that are listening to this, that would be the difference of a muscle cell being like a small diameter circle versus a bigger diameter circle for the muscle fiber or muscle cell diameter itself of your length, not the length of it, but the width of it, if you will. And again, stimulus frequency, this one was talking about wave summation, right? And length tension, that has everything to do with the sarcomere, so that's these pictures here, what it was talking about. So, let's see. So a resting length sarcomere would be the ideal max force produced because it allows the gliding along the entire length of actin versus the other two extremes, which won't be as helpful. If we look at velocity and duration of a contraction, so now you're talking about if you increase the load, so velocity is speed, duration would be how long for the contraction, so how fast can we contract and how long can we stay contracted? So if you have more load or an increased heavy load that you're trying to pick up, your velocity would decrease. You couldn't be as fast because this is heavy. And the duration you're able to hold the contraction would also be decreased. So these are usually pretty straightforward. Just think it through. If something's heavier, can you move it as fast? No. Can you hang on to it as long? No. Versus something lighter. For recruitment, this is increasing the number of motor units. So remember, the more motor units we recruit, the more muscle fibers. You would, of course, increase the speed. You would also increase the duration because you can hold it longer if others are allowed to relax while other ones get recruited, right? And contract. And then muscle fiber type. So this appears a little complicated, but if you think it through, it won't be so bad, okay? So muscle fiber type. So this is talking about a slow oxidative fiber, a fast oxidative fiber, or a fast glycolytic fiber. Okay. So, what do I want to tell you? So all muscle cells have all three types of fibers within a muscle cell. So one muscle cell has all three of those types of fibers. The difference is the velocity and duration of a contraction would depend on the proportion of which type of fibers do you have in that muscle cell. Okay? And with the different types of activity you're trying to do would depend on which fibers are getting more activated than not. Okay? So if we look at this table then, so that's why I put proportion of fibers. All muscles have all three types. The proportions vary due to the predominant type of physical activity. So are you a runner versus a weightlifter? Are you a couch potato versus a weightlifter? Are you a couch potato versus the runner? Are you a weekend warrior and you do all of them? No, right? Okay, so if we look at slow oxidative for slow and fast, oxidative, think oxygen availability. This means like essentially aerobic activity. You are able to do things and oxygen is available while you're doing it. You're not totally out of breath, okay? Versus fast glycolytic. Glycolytic means that you are only being able to break down glycogen, but you can't produce all the ATP by going through later what you guys will learn is the electron transport chain. So because you can't make a whole bunch of ATP, you're burning through your glucose faster because you only get 2 ATP at a time versus if you could do the whole cycle, which you have to have oxygen to do that. So this means this is anaerobic, meaning A, non, so not oxygen available. If oxygen isn't available, you won't make 36 ATP. You guys don't have to know those details of those numbers. I'm just trying to let you guys know anaerobic, meaning like you are, all of a sudden, you had to sprint all the way across campus. You are out of breath. That is anaerobic. Yes, you're alive. Yes, you have some oxygen, but you don't have enough oxygen getting to all of your muscles or the ones that you need at that time. So you're not dying. I'm not talking about that. I'm just talking, and you will cycle through these different phases, which you learn more about in 228. Okay, so all of a sudden you don't have to run so hard. You aren't going to be anaerobic. Now you start breathing normally again. Now you're going to be able to be in aerobic respiration again, where you can breathe okay and talk to somebody and not be like, hold on a second. Okay, that would be anaerobic. Okay, so slow oxidative and fast oxidative means oxygen's available. Fast glycolytic means oxygen's not available. So if oxygen is available, then our ATP use, it means that ATP is available and we'll be able to do the whole electron transport chain and we're making tons of ATP. If it's a slow oxidative fiber, we just use it a little slower. We're not burning through our ATP, we're just doing it a little at a time. And a fast oxidative is burning through our ATP quickly because it's fast and it needs more ATP to do it. Fast glycolytic is burning through the ATP it has, even though it's not going to make tons more ATP because you're out of oxygen. Okay. And let's see. So oxidative, we said, means oxygen. The more oxygen that you have, where do we make our ATP? Who's our powerhouse? Mitochondria. So if you have oxygen available, what I didn't tell you guys was that electron transport chain thing all happens with your mitochondria. So if you have, and you're making tons of ATP, it also means that you have lots of mitochondria making that ATP. And in oxidative, that makes sense that both of them have lots of mitochondria versus the glycolytic, you're out of oxygen, you're bypassing the mitochondria at that point. You're not using him to make your ATP. That's why glycolytic cells have few mitochondria in it, okay? As far as ATP use also would have to do with your resistance to fatigue, right? Because if, let's see, where's our resistance to fatigue is right here. So resistance to fatigue, if you're burning through your ATP slowly, you're not going to fatigue as fast, right? So that makes sense. And if you're fast oxidative, you'll burn through it a little bit faster, so you'll fatigue a bit faster, right? And the fast glycolytic, you're burning through it so fast, you don't have any resistance to fatigue. You're going to feel fatigue. That's why it says it's low resistance to fatigue. Okay? So those all make sense. Let's see, what else do I want to tell you? Capacity to make ATP, that is all tied in with your mitochondria, so that makes sense. If you have many mitochondria, you'll have high availability of making ATP. Same thing for fast oxidative, and it'll be limited because you have few mito, so it'll be limited on your availability of making ATP for a fast glycolytic fiber. So, let's see, capillaries. Here we are, concentration of capillaries. So, capillaries imply what? Blood supply. So blood supply, thus also bringing oxygen to your muscle. So if oxygen's available and mitochondria can make ATP, it makes sense that you would have a high blood supply and oxygen. So what color do you think it will be if you have high of all of those things? Hemoglobin and the red of the blood and red of the muscle, same thing. So that's why slow oxidative are red fibers. Okay. Fast oxidative is, it says lighter red, but you can still consider it a red fiber. It's just, it's not saying it's pink. It's just not quite as red as the other one. So fast oxidative is still red, but look here at our fast glycolytic, they're pale, kind of whitish. Okay. So they have, remember, lack of oxygen. They have less, no oxygen, thus mitochondria they're bypassing mitochondria and they're not making all that ATP. So all of these things are really tied in if you think about, if I don't have this, then I don't have this, this, or this. And that'll help you guys remember who is what. But think about it this way too. If you guys were eating, say, the dark meat on Turkey Day, right? Those would be slow oxidative fibers mostly that you're gnawing on, right? In that muscle. Versus if you did like the breast meat on the turkey, those are mostly your fast glycolytic fibers up there, okay? Because it's white meat and not as red or dark. Some people don't like eating dark meat. I think it gives good flavor, I'm just saying. Anyway. So contraction velocity, if you have to use a lot of ATP or the ATP use is slow, so let's look here. We had ATP use was slow right here. So our conduction velocity would also be slow, right? Because we're not trying to chew through our ATP that fast. If it's fast on fast oxidative, they actually have a fast velocity also. And glycolytic, they're burning through things fast, and that's why it's fast velocity, because they're burning through their ATP reserves really fast because they don't make tons of ATP like the other guys could. Fiber diameter, small for slow oxidative. It just tends to be smaller there. Where's the fiber diameter? Is right here. Okay, and then it's intermediate, so in between length and a fast, not length, diameter, in between diameter on fast oxidative, but fast glycolytic will be the largest one in diameter. And then myoglobin, which is down here, which we said is in our muscle cells to ability to carry oxygen. So this is like hemoglobin. And so the red fibers are going to be higher in myoglobin. The white fibers would be low on the myoglobin. Okay. So that was pretty cool. Effects of exercise on the muscle. So aerobic exercise, we're talking about swimming, jogging, and biking. So you have an increased blood supply and mitochondria. Let's see, what else do I want to say? So depending on what you're asking your muscle cells to do, okay, the muscle cells can change its internal and external environment based on what you need them to do. So long-term activity, not something immediate. It's not like you decided to become a runner just today. Okay, this means like you've been practicing and running and working up to doing it for a length of time, maybe few weeks, few months, that kind of thing. So this isn't a change from today to tomorrow. This is over time. What are you asking your body to do, and how will your cells respond to that? Okay, so if you were doing more aerobic exercise, you're going to have increased blood supply. So you're going to make more capillaries to the cells. So this is an external environment thing. And internally, you would make more mitochondria because you need more energy. If you were a couch potato and decided to become a jogger and you do it over a number of weeks, you're going to make more and more mitochondria because when you were a couch potato, you didn't need all that energy. Why waste your cell resources making mitochondria if you're not going to use it, right? And then more mitochondria means more ATP. Mitochondria has its own DNA, so it can make more of its own mitochondria. Myoglobin is a protein. So inside of your cell, you can make more myoglobin also, okay? Because now you need more oxygen carrying capacity because you are increasing, you need more oxygen to do those activities. So again, this is over time. Not a lot of change in the cell diameter. So the cell doesn't get bulkier looking. And if you look at people that are avid swimmers, joggers, bikers, they're not bulky weightlifter looking people, right? They're usually slimmer. I mean, you could be a heavy set. I'm not talking about heavy set versus a thin runner, but I'm just saying overall, you're not bulked up with your muscle cell diameter. So these guys are slimmer in that regard. So no change really in the cell diameter. For resistance exercise, so this would be people like weightlifting, okay? And again, not because they decided to weightlift tomorrow, but they've been weightlifting over a period of time and changing what they're asking their body to do. The size and shape of the muscle will change because if you use too much force, you could get a muscle strain or different degrees of a muscle tear. And in order for your body to try to prevent you from tearing, it's going to fill up the muscle cell with more myofibrils, more actin and myosin proteins. So that way you're trying to protect it, you'll have a better ability to resist that tearing then, right? And if you do it carefully and properly, you're not just tearing your muscles up when you're weightlifting. Okay, you're going to do it in a slow and controlled way, and then you will actually increase the size of the muscle cell, not the number of cells. So the size is hyper, did I write it on there? I didn't write it on there yet. Okay, I have it. I'm like, what do my notes say? Hypertrophy means the muscle cell gets bigger. So hypertrophy equals muscle cell gets bigger, as in diameter. Another term is hyperplasia. That means increased number of cells, and skeletal muscle cells don't really do that. Okay, so this would be increased number of cells. But keep in mind, muscle cells do hypertrophy, not hyperplasia, okay? So I was going to describe to you what a weightlifter looks like, the difference. I'll have to do that, I guess, next time. So it looks like what I need to do is finish up cardiac muscle or disuse atrophy, cardiac muscle, smooth muscle, and then jump into chapter 11. And so I will make that recording probably by Friday night-ish, Friday afternoon. It depends when the room is available for me to make that. So make sure that you guys see that, or, just watch it sometime before your Thursday exam. The pathways for studying should be available and open. And so you guys should be able to access the study guide and access the learning pathways and the old exams. Remember what we talked about on study habits. Don't hesitate to come and see me later. And then let's do a clicker quiz real quick, and you guys can be on your way.